Okay, let's unpack this. We are diving into a deep, deep paradox today. One that, it really challenges the most fundamental laws of cellular biology.
It really does. We're talking about caspases.
Right, the specialized enzymes famous for orchestrating programmed cell death apoptosis.
And our sources today focus on how a simple fruit fly, Drosophila, rebuilds its wing tissues after massive destruction from ionizing radiation.
So the core mission of this deep dive is to get our heads around something called compensatory proliferation.
That's it, that spectacular, almost miraculous ability of tissues to sense huge damage and then just regenerate themselves.
Yeah.
And we're zeroing in on an enzyme called Dronc.
Which is the main initiator caspase in the fly. So for you listening, it's basically the equivalent of caspase 9 in humans.
Exactly. And you have to remember, Dronc's reputation is, well, it's the cellular assassin.
It gets this signal to kill, and then it kicks off this irreversible cascade. Game over for the cell.
But the key discovery in this research just flips that entire narrative on its head. It shows that Dronc has an essential, non-lethal role. It's actually driving the regeneration.
So the executioner is secretly the construction foreman.
It's the ultimate biological plot twist.
Yeah.
This whole process is mediated by this fascinating balance between two new types of cells they found.
The survivors.
The survivors. The NARE cells, that's Dronc activating, apoptosis-resistant, and the NARE cells, the non-Dronc activating ones.
And really understanding how a cell can activate its own death signal. I mean, literally pull the trigger, but then survive and drive new growth is huge.
Oh, absolutely. This isn't just about fruit flies. It has profound implications for things like cancer recurrence, for resistance to radiation therapy. It's the very mechanism of cheating death.
So where does the story start, I imagine, with the trauma itself?
Precisely. It starts in the Drosophila wing imaginal disc, or WID. The researchers blast it with 20 days of X-rays.
And the immediate aftermath is just chaos.
It's a massive cell loss, just as you'd expect. You see huge amounts of apoptosis. They used markers to see it, and that death signal peaks really intensely around 24 hours post-irradiation.
And the tissue itself must be shrinking, right?
Oh, dramatically. The wing disc is visibly damaged and smaller. It's pure trauma. The tissue has taken a major, major hit.
But this is where the fly model is so incredible. They bounce back fast.
They really do. The regeneration phase kicks in almost right after that 24-hour death peak. Between 24 and 48 hours, the survivors just start proliferating like crazy.
By 48 hours.
By 48 hours, the entire wing disc area is back to almost its original size. It's a textbook case of successful compensatory proliferation.
Okay, so here's the clever part, the methodological challenge. If you want to study the cells driving the repair, you have to be able to find them to tag them.
Right. And the traditional sensors for Dronc activity, they just tag the cells that are actively dying.
Which makes sense. Death is a fast process.
It is. The old rapid reporters, the RDBS, they mostly label the doomed cells. The data showed something like 70% of the cells they lit up were, in fact, apoptotic. That's Dronc doing its day job.
But the researchers needed a way to find the outliers, the cells that got the death memo but decided to ignore it and survive.
So they engineered this brilliant tool, a delayed Dronc activity reporter, the DDBS.
Delayed. Okay, what does that mean?
It's ingenious. Unlike the fast reporters, this one needs the cell's machinery to be fully functional. To glow, it requires the cell to perform transcription-making new RNA and then translation-making new proteins.
Ah, I see where this is going. A cell that's actively dying shuts all of that down, right? The factory closes.
The factory is closed for business. So if a cell activates Dronc and immediately starts to die, it won't have time to make the fluorescent proteins the DDBS needs. It stays dark.
So the only cells that light up are the ones that activate Dronc, but then somehow keep their production lines running long enough to make the reporter.
Yeah.
They cheated the system.
Precisely. And the data was clear. At 24 hours, only about 30% of the cells labeled by this new slow reporter were actually dying. It was proof that the DDBS was successfully tagging the survivors.
The DARE cells.
The DARE cells. Dronc activating radiation-induced apoptosis-resistant epithelial cells. The ones who faced the death signal and, just shrugged it off.
So they're the primary initiators of the repair.
They are. They appear scattered all over the imaginal discs and they start proliferating immediately. By 48 hours, their clones cover almost half the entire wing disc, and they're fully functional. They go on to build normal adult wings.
So what about the other survivors, the ones that didn't light up the Dronc reporter?
Those are the NARE cells, non-Dronc activating. They're the rest of the resistant population, and they're also essential, but they seem to be more like secondary responders, the support crew.
Okay, let's get into the deep mechanics then. This is the core of the paradox. If Dronc is active in DARE cells, but they don't die, how? How do they decouple its functions?
This is really the crux of it. First, they had to prove Dronc's activity was what mattered, not just its presence. So they knocked down the Dronc gene specifically in those DARE cells.
And the result?
A huge reduction in their proliferation. The wing discs at 48 hours were visibly smaller. The regeneration was impaired.
So you absolutely need Dronc to be active, to be, on the job for the construction to happen.
Exactly. But, and this is the incredible part, it doesn't need its tools of death.
What do you mean?
They went in and blocked the whole traditional apoptotic machine that Dronc is supposed to activate. They blocked its partner, the adapter called DARC. And they used an inhibitor called P35 to block the downstream executioners, the effector caspases.
So they cut off the entire death pathway, but only in the DARE cells.
And what happened? Virtually nothing.
Nothing.
No effect.
Well.
DARE cell proliferation was fine, tissue regeneration was fine. This is the definitive proof. Dronc is driving this growth completely independently of its normal death squad.
That is just startling. So Dronc is active, but it's being restrained somehow. It's prevented from linking up with that death machinery. What's providing the restraint?
This is where the unconventional myosins come in. They're the survival switch.
Okay.
The key dilemma is, how is Dronc activated but physically stopped from activating the lethal caspases? And the primary protector they found is a protein called myo-1D.
A myosin, like a muscle protein.
Sort of. It's an unconventional one involved in cell structure, and it physically binds to Dronc. When the researchers knocked down myo-1D in the DARE cells, the restraint failed, catastrophically.
Cells started dying.
Immediately. Effector caspase activity shot up, cells started condensing, full-blown apoptosis.
So myo-1D is like a molecular bodyguard. It's physically holding Dronc back, keeping it away from its weapons like DARC.
That's the leading hypothesis.
Yeah.
That it sequesters Dronc, maybe pulls it away to a different part of the cell so it just can't initiate that lethal cascade.
Yeah.
It's physical separation, not chemical inhibition.
Is it the only player involved in this?
It seems to be the main one, but there's some fine-tuning. They also looked at another myosin, myo-7A, or crinkled.
And what did that do?
Knocking that one down actually reduced the tiny bit of background caspase activity in the DARE cells even further.
So myo-1D is the don't die signal, and myo-7A might be allowing a little bit of non-lethal activity, a bit of a hum in the background.
It suggests that, it shows just how incredibly fine-tuned this whole survival mechanism really is. It's a remarkable system of checks and balances.
Okay, so with survival secured, let's pivot. What are the signals that actually drive the proliferation? Let's start with what the DARE cells are telling themselves, the cell autonomous part.
The central driver they identified is a kinase called P38A. When they downregulated P38A in DARE cells, their proliferation just tanked.
And that affected the whole tissue.
Critically, yes. It led to a significant decrease in the overall wing disc size at 48A. If you take out P38A, the tissue just fails to regenerate properly. It is indispensable.
What about J&K signaling? That's another major stress pathway.
They looked at that too. Knocking down J&K also reduced DARE proliferation, but here's the difference. The tissue was fine.
The tissue was fine. How?
Because the loss of DARE proliferation was fully compensated for by the NARE cells. They picked up the slack. The final wing disc size was maintained.
So P38A is the non-negotiable leader for DARE cells, but J&K is more of a significant but replaceable player.
That's a great way to put it. Now, what's activating these kinases? That's where it gets even stranger.
Go on.
It involves a TNF receptor called Wengin. Knocking down Wengin in DARE cells really impaired their proliferation. But the activator wasn't the usual suspect, the TNF ligand IgR.
So what was it?
It appears to be reactive oxygen species, like radical.
Wait, free radicals, the
Yeah.
things we usually associate with damage. The DARE cells are using their own internal stress signals to tell themselves to grow.
Exactly. They are generating ROS inside the cell using enzymes like DUOX and NOX. And that internal ROS activates their own Wengin receptor, which then drives P38 and J&K, fueling their growth.
So DARE cells are complete self-starters.
They are. But they also recruit help. Which brings us to the non-cell autonomous part, how they talk to their neighbors.
How do they get the NARE cells involved?
The signal that tells the NARE cells to start dividing comes from downstream of that active Dronc and P38 signaling in the DARE cells.
So Dronc isn't just an internal signal, it's also telling the DARE cell to send out a help wanted sign.
It is. And they confirm this by mapping where the dividing cells were. At 24 hours, something like 60 or 70% of all dividing cells were NARE cells.
And where were they located?
Overwhelmingly, right next to the DARE cells. It's clear evidence of a localized short-range signal. The DARE cell shouts, "Grow," and its immediate NARE neighbors are the first to respond.
Now, with all this aggressive growth, you've got to have a brake pedal, right? A negative feedback loop to stop it from going too far.
Absolutely. And that's where the other TNF ligand, IGR, comes back into the picture. When they knocked down IGR, they saw a moderate increase in DARE cell formation.
An increase. So IGR is actually an inhibitor here.
It seems to be. The idea is that IGR is secreted, probably by the growing NARE cells, when it acts back on the DARE cells through a different receptor called Grindelwald to just gently tap the brakes.
So DARE starts the party, NARE joins in, and then NARE tells DARE, okay, that's enough, we're good now. It's a perfect homeostatic loop.
It is, and this balance is absolutely crucial. Let's look at what happens when you disrupt it.
Right, the ultimate test. What happens if you take DARE cells out of the picture completely?
They did that. They used a molecule to just kill off the DARE cells. And regeneration failed. The wing discs stayed permanently small.
So the NARE cells can't do it on their own.
They can't. They are completely dependent on the DARE cells to initiate the whole process. They're powerful responders, but they're not initiators.
Okay, but what if the DARE cells are just struggling a bit, not completely gone?
That's the second scenario. They compromised DARE cell proliferation without killing them. And in that case, the NARE cells went into overdrive. They overproliferated to compensate.
And did it work? Was the tissue size restored?
It was. The overall wing disc size went right back to normal. So on the surface, it looks like a success.
But I'm guessing there's a catch.
There's a big catch. Even though they restored the size of the tissue, disrupting that DARE-NARE ratio messed up the final product. The adult wings that developed were significantly smaller than normal.
So it shows that regeneration isn't just about getting the cell count right. It's about using the right cells in the right balance to build a quality structure.
Exactly. Quality over mere quantity. Which brings us back to this idea of resistance, molecular memory.
The cells that survive, they learn from the experience.
They do. When they re-irradiated the regenerated wing discs at 48 hours, the amount of cell death was cut in half compared to the first time. The tissue as a whole inherits resistance.
And are the DARE cells better at remembering how to resist?
Significantly better. The DARE cells and all their descendants showed a staggering 7-fold decrease in their sensitivity to a second dose of radiation.
7-fold. Wow. What about the NARE cells?
They also became more resistant, but only showed about a 3-fold decrease. So the DARE lineage not only drives the repair, it passes on this highly fortified memory of resistance.
So if we step back and synthesize all this, what's the big picture model for how these cells cheat death?
The model is really elegant. Trauma from dying cells triggers the formation of DARE cells. Those DARE cells, protected by their MYO1D bodyguard, use active Dronc and pathways like P38 to drive their own growth.
While at the same time, they're sending out signals to their NARE neighbors to get them to help out.
And then the NARE cells, in turn, signal back with IGR to say, okay, let's cool it down, ensuring the whole process doesn't run out of control.
It's an incredibly complex, tightly regulated loop that all hinges on Dronc's ability to perform this second non-lethal job.
Absolutely. And when you look at human systems, many of these same players, MYO1D, JNK, P38, are deeply tied to tumor survival and metastasis. These findings are highly relevant for understanding how a tumor might survive radiation and then recur stronger than before.
The same survival switch that DARE cells use to rebuild a wing could be what radiation-resistant cancer cells are using to rebuild a tumor.
It's a very compelling possibility. Now, this research establishes that the whole process starts with a signal from the dying cells themselves. Once the killing has started, the alarm goes up.
Which leaves us with one final lingering question.
The ultimate question. We now know that dying cells initiate this whole regenerative process. But the exact molecular identity of that signal itself, that specific mitogenic molecule, the code red flare, sent up by a dying cell that tells the survivors it's time to rebuild. That remains an intriguing mystery yet to be discovered.
That's the next biological frontier.